So without further ado, I'm going to bring up Casey Pape, who's going to be our speaker. And Casey works with a group out of the Institute of Northern Engineering who's been doing studies on cold-loving bacteria or psychrophiles. And I'm not going to go into that at all because he is the expert on that. But they've been doing some really exciting work with those bacteria and biodigesters that could really make a difference for that technology here in Alaska. So we look forward, Casey, to your presentation. Why don't you come on up? He just got back from Cordova where he's been doing a project there with the students. So he'll be talking about that as well. Casey, it's yours.
Thank you again, Julie, for that nice, warm introduction, and thank you, everyone, for coming to our talk today. Let's see here. I'm going to pull up put down ACEP's presentation and pull up mine. So today, I am going to be talking about our Cordova Biogas project, which is located in Cordova, Alaska. And just a show of hands, is everyone here familiar with biogas technology or anaerobic digesters at all, or kind of have an awareness of what my talk is likely to be about today? Okay, so basically, this project was funded by the Denali Emerging Energy Technology Grant, and this project was titled the Cordova Biogas Project. And my lecture today will be project Energy from Psychrophilic Bacteria, a cold region alternative for biogas. Basically, my talk will go into discussing what our research group is trying to do, and that is anaerobic digestion technology is ubiquitous throughout the world. There's all anaerobic bacteria which consume organic material as a form of respiration in the presence of a non-oxygenated environment, exist in all regions of the planet, but they're most successful in warmer regions. Their biologic rate is highly dependent upon temperature. So what our research group was trying to do was extend that technology further north in latitude, or southern in latitude. So that's what we're going to be discussing today. These digesters are very popular in equatorial regions of the world, but they're not very popular as you go further north and the temperature becomes a limiting factor. So my talk summary will go into the motivating research and the goals of this project and some of the ideology behind it, as well as an introduction to small-scale anaerobic digestion technology. And then we'll go into the Cordova Biogas Project and discuss what we learned, some of the triumphs and the pitfalls of that experiment, and then we'll discuss limitations of the technology and some of the future prospects for the technology in Alaska. And then I'd like to get through that. I would imagine my lecture will go for about 30 to 45 minutes, and then just open it up for a Q&A discussion. And really welcome all feedback, all back and forth. Would be really great to hear what you guys all have to say, and any questions that you have about the technology, I'm more than willing to entertain, as well as my research in limnology and thermokarst lakes for some of you that were expecting a talk that was more along those lines. So the project goals and the summary of the project was to improve conventional anaerobic digestion efficiency through the use of psychrophilic, cold-adapted bacteria found in Arctic lake sediments in Alaska to provide cooking and heating fuel for Alaskan households, again, at the small scale. So really what another goal that we're trying to do is eliminate a waste stream. And this, for example, is a waste stream. This is a Arctic or rural landfill. This was taken by Edda Metters. I'm not sure exactly where, but I believe on the North Slope somewhere. But as you can see, Alaska is usually kind of a final stop destination for trash and one lifetime products. So really we're entering into an age where we can no longer look at materials and products in this kind of linear fashion of a product is made and then thrown away and that's it. We now need to start looking at these things as, even though they have no more use to us, they're still a resource, and we can extract the energy that's still retained in them, or resources that are still available to us. And so organic food waste, human waste, animal waste, these are all resources that we're now starting to reevaluate and realize that, you know, we have to recycle them or dispose of them at great cost to us, but there's actually a resource potential there. So the goal of this research is to try and utilize that for something that we all care about. And in Alaska, that is an obvious one that comes to mind is energy, because we all know that the high costs of energy, electricity, gas, heat, everything is more costly up here due to the nature of how spread out everything is and how much it takes to transport. So this, and this. So the motivating research behind this has really, who gets to take the credit is my professor, Katie Walter Anthony, who's an aquatic freshwater chemist limnologist at the University of Alaska Fairbanks, and a colleague from National Geographic, T.H. Thomas Henry Culhane of Solar City's Blogspot. And he is an urban developer, urban planner. And basically, they became emerging explorers in 2009, and during a meeting, they got to discussing their disciplines. And Katie, my professor, who is studies primarily permafrost and thermokarst topographies, focuses her efforts on studying methane emissions from Arctic lake sediments around the Arctic in both Alaska and Siberia primarily. And T.H. Culhane, who develops small-scale anaerobic digesters in places like Egypt, Pakistan, Nepal, places like that, where the cost of energy is very high and often poor education, poor infrastructure. Biogas technology is coming on very strongly in these regions because it offers great personal benefit and quality of life for very minimal cost. So here's our target molecule. Our target molecule is methane. For those of you who don't know what biogas is, it's a mixture of multiple gases, methane being the primary one, the target one, the one that we're interested in, as well as carbon dioxide and trace other gases, water vapor, hydrogen sulfide gas, and a slew of others, or volatile fatty acids, things like that, but in trace amounts. For the most part, it's methane and carbon dioxide. And this can be burned directly when gathered into a high enough energy density or high enough density, it will ignite. And it can be used for cooking stoves, can be used for heaters, lights, anything that methane can be used for, anything that natural gas can be used for, biogas can pretty much be used for as well. So technology overview, anaerobic digestion, which is the overall process for making biogas, is a multi-step process in which bacteria obtain their energy by consuming organic matter in a non-oxygenated atmosphere or without the presence of oxygen. So whereas you and I breathe air and we're through an aerobic pathway, we release CO2 and water vapor in order to obtain our energy source or to consume sugars and our carbon to make ATP. These bacteria do it in a non-oxygenated environment. And that requires more than just a single microorganism. It requires of a slew of bacteria ultimately. And so the pathway is 3, arguably 4 steps in order to do that. So you have a hydrolysis phase where complex organic matter is broken down into simple sugars, and then from there is fermented, in which acetogenesis occurs, making acetic acid, and then from there it's ultimately consumed by methanogenic bacteria, which release methane as a byproduct of their respiration. So these are methanogens, and methanogens are defined by the temperature range in which they occupy. So there's three main classes of methanogens, thermophilic, mesophilic, and psychrophilic. And what this project focuses on is the psychrophilic. Thermophilic and psychrophilic are both labeled extremophiles in the fact that they live in environments that is typically assumed to be non-ideal for life. So they're labeled as extremophiles because they live in extreme climates. And that's all I'm going to say about that. I've got a little picture here of methanogens, a type of methanogenic bacteria. This is a colony type bacteria, but I want you to know that they exist in all different forms, single-celled organisms, flagellated, mycelium, all sorts of stuff. So that they don't look all just like that. So basically, what we're trying to mimic in this experiment is like a cow's stomach or your digestive tract in non-oxygenated environment where bacteria can break down organic material and be cultivated in a warm place, typically warm place, in order to utilize organic material and release methane. So we're trying, since our goal is the methane, we want to try and optimize the conditions in which those bacteria live. And the guiding research, what my lab group mainly focuses on is these thermokarst lake depressions in and around the Arctic. And these same bacteria that can be found in cows' stomachs and yours and my digestive tracts are also found in benthic lake bottom regions in the Arctic. There they are experiencing much colder temperatures than you or I exist at, and certainly what our internal bodies are providing for these bacteria via homeostasis. So yes, if you have any other questions about that research, feel free to ask as well when it comes to the Q&A. So the basic design of an anaerobic digester, which is what we built in this experiment, is very simple. Since we're ultimately mimicking a stomach or a rumen, there's really not too much that you require in that. You just need to have a non-oxygenated environment, so no exposure to air. You need a warm or wet place, and you need to be able to collect the gas that comes off of that. So this is a very simplified model. This is the basic RT style. RT is a appropriate rural technology institute based out of Bombay, and they specialize in making rural adapted digesters. They can be kitted up, they can be shipped out, they're appropriate for regions where you have limited access to materials, and they're very minimalistic. At the industrial scale, there's really not a whole lot of difference between small scale and large scale. It's just there's more mechanized inputs of mixers, of heat exchangers, all designed just to kind of optimize the reaction that's going on inside the vessel. But basically, you're just trying to keep a wet space in order to harbor bacteria and a carbon source. And the point of this slide is to emphasize that there's multiple ways to get to grandma's house. There's no one style that's patented, or this technology is absolutely accessible to anyone. With just a few components, with a few tools, anybody can make one of these. But there are some things that need to be considered before attempting it on your own. So this brings us into the Cordova Biogas project. And the Cordova Biogas project is located at Cordova High School, or directly adjacent to Cordova High School. And it's a cooperative research effort between the University of Alaska Fairbanks and Cordova Schools, Cordova School District, and Cordova Electric Cooperative, CEC. And so the project is two-phased. In the first phase, we are doing more of the science, just trying to find out what are the optimal conditions in which we can provide these bacteria at colder temperatures, and what can we expect as far as a yield from that. And then the second phase, which is to try and utilize and demonstrate that technology to Alaskans in order for them to kind of get their head around the technology and see how it might apply to an individual's life or just utility. So in the first phase, we built and deployed 6 1,000-liter digesters, which were recycled sorbexol, or sorbitol, sorry, sucralose containers that were used for like canneries and fish processing facilities in Cordova. And then within that, we inoculated each tank with a different slew of bacterium. The bacteria cultures were either obtained from farms in Delta Junction, which was essentially collecting a bunch of cow manure and putting them into the tanks and then putting in food waste, or we took lake mud samples from Goldstream Lake in Goldstream Valley in Fairbanks, and those were that was basically the amount of it. We separated the tanks into 2 separate rooms and isolated them temperature-wise. One room was labeled cold room, which isn't really cold, but 15 degrees C, and another room which was 25 degrees C. And conventional digesters shut off around 25 to 30 degrees C. 30 degrees C is kind of the minimum that is required for small-scale digesters to really work well and effectively for an individual household or otherwise. And so anyway, we wanted to test temperatures that were slightly less or non-ideal in order to see whether or not we had anything going with these psychrophilic bacteria. So as you can see, tanks number 1, I'm just going to label them off: 1, 2, 3, 4, and 5, and 6. Tanks number 1 and 4 were psychrophilic cultures only. Tanks 2 and tanks 5 were a mixture of psychrophilic lake mud and cow manure cultures. And then the tanks 3 and 6 were just strictly mesophilic. So the idea here being we would see a distinct difference between tanks number 1 and tanks number 3, tanks number 4, tanks number 6, and then possibly some sort of displacement of one population over the other in tanks 2 and tanks number 5. So a summary of the data, and I'll just go through this and we can talk more about it during your questions, but we monitored temperature, we monitored ORP, we monitored pH, we monitored dissolved oxygen, but ultimately we monitored biogas production. That's what we all cared about. So here is a section of the data from this last year, and short answer of why I'm only presenting a section is because we had numerous issues with biologic rate and trying to get the data to be presented in a useful way, and the long answer is it's quite long. So anyway, this is a fairly useful graph where you can see that the warm rooms, it's a clear distinction between warm room and cold room. At 25 degrees C, the bacteria did much better than they did at colder temperatures. And in fact, only one bacteria culture was observed to be producing any gas at all beyond nominal amounts in a 15-degree environment. So the response curve for temperature versus biogas production for anaerobic digestion is kind of a hockey stick or exponential growth model. It falls off very sharply about 20 degrees C and is almost nominal below 15 degrees C. And we've essentially demonstrated that that is in fact true. But we did observe that psychrophilic cultures were more efficient than mesophilic cultures in any one example. So ultimately that is pretty encouraging. But for similar scale and size reactors of this type, at 30 degrees C in India and China could be expected to produce as much as 1,000 liters per day, which is enough to provide a single family residence with heating or cooking fuel at least. And here, our greatest efficiency was less than a third of that. So anyway, these are the digesters that we have. We located them in size of a 40-foot connex and insulated both rooms. And then you can see in this picture here that there's a positive pressure on each of the tanks that are producing. I guess that is the point of that slide. We did observe a very noticeable acidification among all of the digesters. Once we sealed them off and first inoculated the bacteria and started feeding, that was attributed to overfeeding in the initial part. It's well documented in the literature that acetogenic bacteria, which occur before the methanogenic pathway, can outpace essentially methanogenic activity. So for anyone interested in starting up a personal digester, it's really important that you're trying to pair those up as closely as possible and get them to work symbiotically and in line with each other. Because once you have kind of a runaway acetogenic effect, the pH goes down, and methanogenic bacteria are very sensitive to acetogenic, or are very sensitive to pH. So they will effectively shut down and stop producing methane. So what we did essentially is we shut them down about mid-March of last year and waited for them to rebound. And in some cases, we had to treat them with lime and with sodium hydroxide in order to get their pHs back up before they could start producing methane again. And so this will take us into phase 2 of the project, which was to try and demonstrate practical household uses or practical uses of this technology at the household scale in Alaska. And so we had a bunch of different kind of ideas about how to do this. There's all sorts of devices that you can run off of biogas. Like I said, it works pretty much just in place of natural gas or propane. But it's important to think about you're ultimately energy limited because you have only about 1,000 liters a day to deal with, or in this case, about 350. So what can you really do in a practical manner? So this brings us into the student involvement, which I, from my own personal experience, thought was one of the greatest benefits that we learned from this project, and that was pairing this university research with a high school institution was really a rewarding experience, not only for the grant, not only for the students, for myself, but just to the whole process in general. This project really couldn't have taken off without student involvement. They were involved with every step of the leg. They helped with construction. They did daily monitoring, daily feeding of the project. They presented and really were outspoken in presenting and discussing the grant. Really kind of the rock stars of the first round of the Emerging Energy Technology Grant. So really was really impressed with their efforts here. They met with the state senate. They had all sorts of really cool, innovative ideas to pair this technology with greenhouses, to test and find ways to purify biogas by, here's a picture, them bubbling it through lime water and measuring the CO2 content on either end. So just really impressive bunch of students that I got to work with. And it really goes to show that they were just really excited to be involved with something, because in rural communities, you know, they don't often get too many opportunities. So it was really kind of awesome to see the university take stock in them, their electric cooperative to take stock in them, and then see where they went with it, because they really ran away with it. So a couple of quick demonstrations. We had to find a way to store the biogas, and we did so by sleeving it inside of this big 1,000-gallon tank, mimicking the RT style gas collectors. So it's kind of hard to see, but there's actually a tank inside of that tank, which is used to passively store and deliver the gas for powering devices. And so here we have a pot of boiling water, just as a clear demonstration. And in the lower right-hand corner is a generator that we got to run for over an hour on about 300 gallons of gas. So just directly in line. And forgive me, I will try and show you some. But some of the students had some cool videos.
I'm Ben Americus, and this is my colleague James Allen. And we have an inner tube full of methane that we collected from our biogas digester. And we got this little cord coming here, connected onto this other cable, and we electrotaped it shut. Then we got another cord right here that comes into the fuel intake. And that's shut closed, so hopefully it'll block all the gas from leaking. And I guess we're going to try to get this to run right now.
Yeah.
Holy dude! Holy dude! We're running on biogas! Woo-hoo!
So they were obviously excited about it. Sorry about the video here. And then I have another one. I'm not sure if it'll do the same thing on us, but this is running a generator on biogas, which was as simple as plugging it in. And I'll let the video show, and I'll explain it as it goes. So this is a Husky 1850 watt gasoline generator, 4-cycle, and all we did here was just plumb a line into the carburetor, and had it running at about half psi. It kicked over, so a little bit of starting fluid, and that's all it took to get it going. We're at really low pressure here, less than a psi. And like I said, this ran for just over an hour on about 300 gallons, so right around 1,000 liters of biogas. Okay. Going back to the earlier graphs that you saw, there's some obvious limitations of the technology. For those of you thinking you have an interest in the technology, there are certainly some things to consider before adopting one of these. Otherwise, you probably just end up with a frustrated mess. So the first one is that it's a very temperature limiting reaction. And we were unsuccessful in demonstrating sufficient biogas production at suppressed temperatures that you could be expecting to encounter outdoors in Alaska. So first and foremost, if you were interested in a small scale, you're going to have to figure out a way to heat this in order to get any sort of BTU output from a digester. There is an odor concern with these, though we did notice that working with lake bacteria, it was very interesting. The tanks that were using cycrophiles smelled like a pond. Still not the most favorable smell, but not nearly as bad as something that you would expect from a manure-based digester. But anyway, it can be a very mechanized, hands-off process if you design it right, but that's something to consider. If you don't particularly enjoy the smell, you need to figure out a way to not handle the material in a very high exposure type setting. The labor inputs can be substantial if you don't consider that when you first design your project. You need to find a way to try and get it, like I said, as hands-off as possible, to grind up your food and make it available to the bacteria in a way that doesn't require much physical labor or input in that sense. And then it's susceptible to winter freezing conditions. So like you saw with our gas collector, actually inside of that collector was about 500 gallons of water first, which is used as the air seal to keep the gas from escaping to the environment. And that has an obvious issue for winter ice conditions here in Fairbanks or anywhere in Alaska, including Cordova at the time. So you will have to find a way to either insulate it, or there's other types of ways of storing and collecting gas. But ultimately, your reactor has to be an aqueous solution. So you have to find a way of keeping it warm in the wintertime, or just starting it up seasonally, if that's your aim. And then additional benefits should be considered beyond just a BTU rating, because these reactors have all sorts of applications for primary water treatment, digestion of pharmaceuticals, of all sorts of things. So there's all sorts of other benefits of these reactors beyond just that of producing gas. So really, where these are starting to come on strong is in areas of high organic waste streams and pollution into the environment. So incidences of fecal coliform, if they're an issue, installing anaerobic digestion units in your dairy operations around your area can severely limit that or reduce the amount of incidences there are with that, because you're focusing the waste, you're treating it locally, and you're reducing the nutrient and bacterial release into the environment. So the current trends in Europe and the rest of the world, these are being adapted with increasing pace and scale and immensity of the operations. In the United States alone, there's now over 300 recognized biogas plants in the United States. Mostly are located on dairy farms, because there's an obvious train of thought there. You have a really mechanized, concentrated location where you can process the animal waste and focus your efforts, whereas being really spread out kind of makes that more difficult. But nonetheless, it's becoming very apparent that we need to start looking at these waste streams as more of a resource and figure out a smarter way to deal with it than just release it into the environment. So at the large scale, future of the technology for Alaska is likely to be more adopted into the large scale. For example, capped landfills is a good example. There you have a product called sour gas, which is more or less the same. It's slightly different because it has higher sulfide content and hydrogen sulfide. But here in Fairbanks, we have one capped landfill with 13 plugged wells. They're not doing anything, but this summer they're likely to start research on feasibility of tapping those wells. And in Anchorage of this year, they're going to be doing the same. They're actually going to be, they've got an active flare in Fairbanks, and they're going to be installing all the wellheads, or they're going to be installing the equipment to harvest the gas from those areas. And then here we have biogas in Kirstenstad, Sweden, which is a similar cold environment, certainly not warm by any means, and they have a highly mechanized, highly industrial scale processing facilities all throughout Sweden. And they've got some really cool projects where they kind of integrate the technology into all facets of life. So they take in slaughter waste, they take in all organic waste from individual residences and households, and they process it and produce hot water, which they can use to regulate the digester itself and increase its efficiency. They can sell that back onto the market. They can use the gas for public transportation in buses and trains, and they can sell the effluent from the digester as a commercial fertilizer. So there's really a zero waste stream here, and it's just a matter of being smart about how you dispose of of the waste and make it into an actual viable product. There's the Vanderhoek Dairy in Linden and BioTier systems in Oregon, as well as many different projects. The BioTier project in Eugene, Oregon is actually kind of interesting because it's noted as a more psychrophilic type reactor where they're cultivating bacteria around 20 degrees C. So there actually is some potential for psychrophilic bacteria at the large scale, and there's no reason why it shouldn't be. So our project has been really successful, and I give a lot of the credit there to the students' involvement. We've been featured in quite a few different press releases and noticed in the media, so we've gotten quite a bit of coverage, and that's been awesome. And we are actually going to be producing a flyer through ACEP here soon, and it's in its final. This year? Oh, it's here. Okay, so it's in the back. Anyway, really excited about that. That's a great little handout for those of you just interested in a summary of the technology. So with that, I'd like to thank you for your time, and I've got all sorts of time for questions or any sort of discussion, so thank you. Yeah. The upper limit? Well, it really depends on the type of bacteria that you're catering to. For mesophilic reactors, it's typically about 35 degrees C to 50 degrees C. And then for thermophilic reactors, which are the most efficient, you know, kind of makes sense, highest temperature, most efficient, those operate in a range of around 56 to, I believe, about 60 degrees centigrade. And then there, it falls off again. Oh, these? Psychrophiles go up to about 25 degrees temperature, and then they just probably are replaced by mesophilic cultures. But the end goal here, or the end message is the warmer the better. They all do better at warmer temperatures. Oh, before you start killing them? It's usually, like I said, it depends on how fast you change the temperature. They're all pretty sensitive to temperature. So if the rate at which you change the temperature is by about more than 5 degrees centigrade in an hour or so, you'd probably kill all the culture and have to start anew. So the upper limit, I've only seen it go up to about 25 degrees C, and it keeps going. So I don't really know what they're tolerant up to until they just die.
You also said it was freezing. I think that if they were done on lakes, you could actually freeze them and eventually thaw them out. What happens in the spring?
Well, right, but even then, the sediments are kind of being protected, and for the most part, you're right. They hibernate for the most part. But the rate, since we're only really investigating the end product, methane, that's not necessarily determining anything about whether or not there's still live bacteria there. It's just that process, they typically hibernate and they won't produce any more biogas, or they won't produce any more methane. And if that's your only way of really seeing if there's any biologic activity there, then for our purposes, it. Oh yeah, absolutely, and again, it only takes, you know, a small culture to ultimately become a large culture. Yeah.
There's a lot of waste heat.
Yes.
And have you thought about capturing waste heat? Let's say someplace is burning diesel fuel.
Mm-hmm. Mm-hmm.
And the exhaust is just going out into the atmosphere.
Yes.
And it's pretty warm.
No, that's a.
Before you use that to heat your digester.
That's a perfectly good example of how this technology can succeed in Alaska. If you pair it with other waste streams, or if you pair it with for example, if you're able to find a waste heat source, then you know, it's got other additional benefits that could really justify it. But if you're just looking at it from the traditional linear sense of, you know, how many BTUs does this have, and from that lens, then, you know, it's hard to justify the technology just as a standalone technology. But if you compare it with other things, then it makes a lot of sense. And so, like I was kind of touched on about the Kirstenstad, Sweden example, they pair biomass burning with biogas generation. They like the biogas for its clean burning fuel source, so they can use that in public transportation. And so they'll burn a lot of biomass and use hot water from that to heat their reactors, and then they get a really pure, high quality fuel that they can use in other areas where you don't want to use biomass, or that's doesn't really make any sense. So you're absolutely right. In Cordova, we had it adjacent to the building, but you could easily see in a high school setting where this is kind of science in action, a cafeteria could be recycling their food waste and processing it in our boiler room where it's quite warm, and use the gas that they have to be, you know, fuel on their Bunsen burners on their lab benchtop or something like that, you know, and it would help students kind of connect the dots as far as where their energy is coming from, what it is, and, you know, there's all sorts of other ways of looking at this. But that's a very good point. Yeah.
Did I hear you right that the cold temperature bacteria were more efficient?
Well, certainly from.
Casey, can you repeat the question?
Oh yeah, I'm sorry. The question was that were the cold weather bacteria more efficient? And it's certainly at the colder temperatures it would appear so. Around 15 degrees C, only psychrophiles were the ones observed producing methane, and then even at 25 degrees C, which is still not ideal for mesophilic, which typically is around 30 degrees C, the psychrophiles did do better. But even then, it started getting pretty close around that.
There's a question in the back.
Yeah, yes.
The cold temperature, the cytophiles that produce the methane in these thermokarst lakes, are they operating at something, I mean, I presume barely above 0 degrees Celsius?
Correct.
They're able to generate a fair amount of methane.
Yes. And the thing there is oh, I'm sorry. The question is in thermokarst lakes, in Arctic lakes in the wintertime, the water temperature, even towards the lake bottom and the sediments, is right above freezing. It's very cold. And here we still see large deposits of methane that can be found at the bottom of the ice or still suspended in sediments. And the real explanation that I have for you is they experience a sharp drop-off in efficiency as well. But there, they just have a lot more time lag, or they have a lot more time to consolidate more gas, and they have much more extensive networks of bacteria that go into the sediment. So you're essentially pooling a very little amount of gas from a very large area, and then that's being expressed only. Oh, absolutely. Yes, absolutely. Mm-hmm. And even we find that the gas composition in the wintertime has less methane content in it than in the summer.
More CO2?
Yeah, more CO2. But that, I really shouldn't get into that discussion because that's kind of what we're working on right now, trying to figure out why that is, if that's decomposition in the water column or what. So that's a kind of an unknown.
Is there another question in the back? Okay.
Yes.
I'm wondering how you use these findings of this bacteria working in cold conditions to improve the efficiency of a compost pile. I mean, this is not, the main object is not getting methane, but it is keeping this heap of compost
Right.
And I'm wondering, is anything that can be done to improve the condition of that compost pile to encourage this cold bacteria?
Well, you raise an interesting point there, because essentially in a compost pile, you're talking about an aerobic decomposition of material. And in fact, you and I breathe oxygen and we're very successful. And as it turns out, it's a much more energy efficient way of decomposing organic material. So if you notice, your compost pile is typically very warm in the center. It actually gets a lot hotter than you would ever expect. It's much more exothermic process than anaerobic digestion, which is what we're talking about here. So that brings up an interesting point in that you could potentially use compost piles and compost in order to insulate and heat a biogas digester and then increase its efficiency through that pathway. Using it in the reverse, I don't really know how you do that. But my background slide here was Chino Hot Springs, expressing that there are places where there's excess thermal, which could be used in order to help process waste. And there, Bernie Carl has said blatantly that he's just burning a tank of propane in his greenhouse in order to try and increase his CO2 fertilization for his plants. And here is an example of, well, you could burn the methane that you're generating in a biogas plant or an anaerobic digester to increase CO2 fertilization in the air, reduce methane release to the atmosphere, all sorts of things. Pick your favorite. Does that answer your question? As best as I can, I guess. Yeah. Yes?
Can you discuss a little bit what the students in the Cordova experiment used for their cultures, for their cycrophiles and their mesophiles? Sources of the cultures?
Yes, yes. Basically, we took our cultures from two places in Alaska. The first was from a lake in Fairbanks, so we cored lake sediments in Goldstream, and I believe Goldstream was the only lake that we took from. And that was just straight lake mud that we used, and we just threw it in these tanks and started adding food to it and water, and methane started being generated. And then the other mesophilic cultures were being obtained just from cow manure from dairy farms in Delta Junction. So
those were just naturally present in the digestive tract?
Yes, in cows, in you and I, in everyone. So typically in China and India, that's what's used to inoculate their tanks, is just manure. And then, yeah, I guess manure is the best source I can think of.
So it looked like tank number 5 was the most productive. That was a combination of cycrophiles and mesophiles, is that correct?
Yes, and there, it's kind of interesting. It's hard to really make too many assumptions about, can you see, about what's happening here, because it's a mixed culture, and we don't really know whether or not one is outcompeting the other, or if they're both just doing well at that temperature, or what exactly it is. So what we found here is that indeed, yes, 5 was the most productive, but we also had some issues with sampling and kind of overflow of material. Sometimes our vessels would get overpressurized and overflow, and so you could lose some material. But for the most part, that's all within a range of, it's pretty much the same. Though I don't want to say that definitively, because we just don't know.
It looks like tank number 4 was very close.
Mm-hmm. Mm-hmm.
And that was composed entirely of cycrophiles.
Entirely of lake mud, yes.
Casey, can you speak to the timeline as far as from inoculation to methane production?
Yes, I can. So these tanks were sealed as of February 19th, 2010, and they started producing flammable amounts of methane 2 days later on, I believe, January 21st. And after that, they kept producing. We didn't have the same instrumentation in place for measuring, so unfortunately our data set is in some places absent or just not as well understood. But what we started finding pretty shortly after that, within 2 to 3 weeks after that, was a very noticeable decline in pH. And so what that was attributed to is we had students who were feeding on a daily basis of 1 kilogram of food and 1 kilogram of water into tanks that really didn't have that much of a biological or activity in them yet. They didn't have that much of bacteria cultures inside. And so what essentially started happening is acetogenic bacteria, yeast, things like that, started fermenting the material more and causing a decrease in pH. And that has been documented throughout the literature to substantially hinder biogas production, or at least methane production. So your first indication when you start to see a decrease in pH is to stop feeding. And so we stopped feeding for about 3 or 4 months until June of that year, at which time the pH was increased back to around neutral pH. So anywhere from about 6.5 to 7.5 is an ideal range for methanogenesis and biogas production. And so once they got back to an acceptable range, then we started feeding in them again, and started feeding again consistently in September of this year, at which time I came onto the project. I've been on with the project since September of this year, and we've been feeding consistently ever since, and really started noticing substantial biogas production above 100 liters per day in December and January of this year, or last year this year. Does that help? And then we stopped feeding in mid-March, and at which time I relocated to Fairbanks, and we have just been kind of leaving the reactors to sit fallow. Yes?
When you say feeding, are you continuing to give them the same sort of material that started them, or is it food?
No, after the initial inoculation of bacteria, we just fed strictly food scraps, and we pretty much pasteurized that material each day by freezing it beforehand, and then we'd remove it, let it heat up, and mix it up, and then put it into each of the digesters. But it was just recycled food scraps, so it was whatever they had at the time. And we have a catalog of all the samples that we've taken from what we fed, but it was a mixture of all sorts of things. But in general, what you would like to feed is the energy-dense materials. So, carbohydrates, proteins, fats, things like that, as opposed to like vegetable waste and cellulosic material.
I know experimenting on changing the types of things, because I think you can affect the pH just by what you feed it.
Oh, certainly. Yeah. I mean, if you feed a lot of yeah, right. Cellulosic material generally around the board doesn't get decomposed that well. So, but it can actually be helpful as space, or the surface area of it can be used to colonize bacteria, so it can be helpful to you. But in general, carbohydrates are known to get consumed by acetogens pretty quickly, and so that can decrease your pH relatively quickly. Like I said, if you can aim for the high energy dense materials, you're in pretty good shape. So if you have a lot of fat, a lot of protein, stuff like that, it's better than less energy dense material.
There's a question over.
Yes?
What are your chances of getting methane from your septic tank? There are a lot of septic tanks.
Oh, I mean, I feel like one of these vessels works as a perfect exchange for one. Your septic field, I don't believe there's, I don't know too much about septic systems, and if you can recover the gases from them, because aren't they kind of permeable drainage fields? But
Usually you have a tank
mm-hmm.
where there's digestion that takes, hopefully digestion takes place.
Right.
But in this climate, it doesn't.
Right.
You basically restore, you strain the liquids, the solids out of the liquid.
Mm-hmm.
The liquid goes into the drain field.
Right.
But you do have at least 1,000 gallons or more
Mm-hmm.
of material that's there that in a warm climate would be digested. In this climate, it just gets stored in your septic tank.
Right.
So there is a collection.
Mm-hmm. Mm-hmm.
Everyone, there are vents. You have to have vents on your septic tank just to let the methane and other gases.
Right. So that's an example. In China, especially right now, they're starting to develop whole projects with sealed anaerobic digestions to process all of the sewer, gray water, black water that comes off of these projects, and then is recovering the energy from them. So if you had a sealed vessel for a septic system, it's not a big stretch of the imagination that you could siphon off and use the gas from that. There, you might be better served by having some sort of heat exchanger that went to your septic tank in order to stimulate the bacteria. But you've also got, I mean, these bacteria are fairly robust in the amount of material, or the types of material they can consume. Salinity isn't a big issue. They can digest soaps and detergents just fine. So, yeah, I think there's umpteen amounts of potential for this technology. It's just been kind of ignored for quite a while. And you just have to be clever about how do you do it. Yes?
Would there be a point where your production eventually dropped off and you have to unseal the thing, clean it out, and re-inoculate it, or can you just like...
Yes, that does happen. We never encountered that during this project. They always seem to be doing really well. And in fact, I just got back from Cordova, and for the past month or so, month and a half, we weren't feeding them at all. And typically what you'll find is we had an event where our tank number 1, our colder room, pH was starting to decline. It was down below 6.5. It was about 6.1 to 6.3. And so we stopped feeding it, and it just stayed there for quite a while. And then that's where I left it in March when I moved to Fairbanks, and then going back to it in June, it had rebounded all the way back to about 7.0 in pH. So that shows that it's a whole consortia of bacteria, and really you don't have any one bacterium. So you can really see that there's kind of a buffered solution in there, or at least the health of the tank can get better and get worse with time. But in general, you have a fair amount of time to play with these things before they get bad. But yes, in industrial scale, there's lots of different types of reactors, all good and bad. They have all their own good and bad things about them. But there you do see, if you're not careful, you can get them to crash, and there they have to do the messy job of completing them completely out and starting from scratch. So it's good to design them in a way so that it's easy to do that when you have to do it, and it's easy to start it back up again. But if you need to drain it, you want it to be set up so that it's easy to do that. Anyone else? Okay, thank you very much.
Thank you, Casey.
Yeah, thank you guys.